My name is Eric Lader. I'm the Senior Director of Product Development here at Qiagen. It's my pleasure to present this webinar to you today. This is the part one of a four-part webinar series that we're providing this month, basically on microRNA and its role in human disease. Webinar 2 and Webinar 3 will both be given by Jonathan Schaefer, who's a research scientist in my group. Webinar 2 is a broader introduction to microRNA biogenesis, function, and analysis. Webinar 3 is a much deeper dive into microRNA expression analysis software tools that are used to look at microRNA expression. And Webinar 4 is advanced functional analysis of microRNA. So this is how to use mimics, inhibitors, target protectors, and so on. So before we go any further, I need to mention that the products described in this webinar are intended for molecular biology applications. These products are not intended for diagnosis, prevention, or treatment of disease. So what is a biomarker? A biomarker is a characteristic that can be objectively measured and evaluated as an indicator. An indicator of what? An indicator of normal biological processes, pathogenic processes, or pharmacological responses to therapeutic interventions. And so the other word that's thrown around with biomarker is the word surrogate. So a surrogate biomarker is a biomarker where you're not actually measuring the disease, but measuring a marker of the disease. So for example, if you're looking at diabetes, you measure sugar level in the blood, and that's a proxy for diabetes. Or enzyme levels from liver or enzyme levels from kidney are a proxy for liver disease or kidney disease. And there's a number of technologies, essentially any technology can be usurped for biomarker discovery, including those that look at the genetics of the situation, protein arrays, sequencing, in situ hybridization, and so on. However you gain access to these molecules is the tool that you use. One more term, a non-invasive biomarker. So in order to use a biomarker for routine diagnostics, the sample material must be as easy to obtain as possible. So what do I mean by this? Well, if we look at our smiling faces here, the supreme material for a biopsy, for biomarker development could be urine or saliva. These are samples that are very non-invasive in terms of collecting from patients. Moving down the scale, a drop of blood from a finger prick, for example, like those that diabetic patients use to monitor their sugar levels in their blood. It's a little bit more invasive. Blood samples are generally the gold standard. They're routinely taken from people by technicians. And then we get down to cerebrospinal fluid and even more invasive surgical biopsies. So all of these materials can be used to develop biomarkers, but the non-invasive ones are clearly desirable if we can move that direction. And from blood, we know that there's multiple components, and from either whole blood or serum plasma or buffy coat or red blood cells, we can look at nucleic acids, proteins, lipids, and specific cell types like PBMCs, endothelial cells, stem cells, or circulating tumor cells, any or all of which can be combined in order to develop a biomarker panel. Another term, cancer biomarker. So when we think about disease, in particular when we think about cancer, you realize that there's a number of different applications for biomarkers in looking at human disease. So for example, there are biomarkers that are prognostic. So is this spot on my arm likely to develop into cancer? So, or is this a serious cancer? So there are value to predicting whether something is going to be bad for you or not. A diagnostic biomarker is more towards the specific type of disease that you have. So a diagnostic biomarker, for example, would be when you have a tumor on your liver, which may not necessarily be a tumor, a liver tumor, but could possibly be a metastatic breast tumor. This would be a situation in which you'd like to use a diagnostic biomarker to figure out what kind of cancer you have. Predictive biomarkers are moving into the realm of personalized medicine, and they're designed to answer the question of whether, for example, a particular drug or treatment is the optimal one for my particular cancer. And so obviously you would be looking most likely at the actual cells from a biopsy. Pharmacodynamic biomarkers would be designed to help the doctor decide what the optimum dose of a drug would be for you, and there are a number of these on the market as well. These will also fall in the realm of personalized genomics and personalized medicine, because obviously everyone is different in terms of the complement of genes that they have. And finally, other prognostic biomarkers where you can look at a tumor or cancer or any disease and ask, well, what's going to happen in the future now that I've been treated? Am I going to relapse or not? So you can see that I consider that these are all sort of in the realm of personalized medicine, because they depend upon your genetics and your epigenome, and the prognostic and diagnostic biomarkers are more generic. So let's talk about circulating microRNA. And what I'd like to cover is an introduction to microRNA and disease. Why do we want to look at microRNAs? Where do we access this information? And as I mentioned, the least invasive would be urine. And we're also going to talk about blood and cerebrospinal fluid, but any other body fluid is a reasonable thing to extrapolate to in this talk. We'll talk about initial screening considerations. How many samples? How do we define our sample population? How large of a library are we screening? What platform are we using? And then I'll talk about candidate selection for potential biomarkers, how we do normalization, some perils and pitfalls. Moving to secondary screening, where we're doing cross-validation using a more focused panel of assays that we've chosen from our primary screen. And ultimately, what you'd like to do is validate your performance using this highly developed panel of assays and collect clinical samples outside of the training set to see how well your algorithm would work on naive samples. So microRNA is transcribed by Pol II in the nucleus as a long primary transcript. That primary transcript is very similar to the primary transcript of an mRNA. In fact, there are microRNAs that are contained within the introns of mRNAs. There are long primary transcripts that also contain multiple microRNAs that are spliced out. So in the nucleus, this prime microRNA is recognized by DROSHA, and what it recognizes is the stem loop structure of a precursor microRNA, and DROSHA will then clip the excess, essentially, transcript off, leaving you with this long hairpin precursor. That precursor is then exported out of the nucleus through an active transport system that uses the transmembrane protein exportin 5. Once in the cytoplasm, this pre-microRNA is recognized by Dicer, which then cleaves it up even more into this mature microRNA. And the hallmark of a mature microRNA sequence is a mostly complementary duplex of around 21 bases. You can see that the hairpin is cleaved off here. This gets physically handed off to the RISC complex. RISC has a helicase activity in it, which unwinds these two strands of microRNA, and it can incorporate one or the other strand into the complex. And for some loci, both strands are used as microRNA, and some loci, only one strand is used as a microRNA. This programmed RISC, this RISC that contains this strand of microRNA, this guide strand, then seeks out regions of complementarity in mRNAs. If we find a perfect match, RISC can actually cause site-specific cleavage of an mRNA. If we have a partial match to the microRNA sequence, and the rules of such will be talked about by Jonathan in the next webinar, then you can have translational repression and mRNA degradation. So regardless of what happens here, the end result is that you have a lower level of protein made from this transcript. Let's talk a little bit about the microRNA timeline. It's a fascinating thing. This has all happened in our lifetimes, or at least most of our lifetimes, and things really didn't pick up incredibly fast until maybe 12 years ago. So in 1993, Victor Ambrose discovered the first microRNA, which was Lin4. It was an oddity. It was a functional small RNA. Nobody really knew what it did. in 1998, Fire and Mello discovered that long double-strand RNA, if you essentially feed it to C. elegans, can be chopped up into small bits of RNA, and those bits can silence genes that have homology to the exogenous RNA that you put in. They also found that there were naturally occurring small RNAs in C. elegans that could do the same thing. David Balcom's lab showed that this happens in plants, so it was more of a general phenomenon. Gary Rufkin in 2000 found the second naturally occurring microRNA, LET7, which is grown to be very, very important in terms of cancer and other diseases. And Phil Zamore's lab, Clone Dicer, showed that you could take double-strand RNA and put it in an extract from flies and so on, and make shorter microRNA. In 2001, a number of labs showed that through cloning and sequencing that there were large numbers of microRNAs in a variety of different types of species. And this was Dave Bartel's lab, Tom Tuchel's lab, and Victor Ambrose's lab. And so here we're looking at, wow, maybe we can use microRNA as a tool. And the real breakthrough here was in mid-2001 when Tom Tuchel's lab showed that if you take small RNAs rather than long RNAs and transfect them or transfer them into mammalian cells, that you could bypass the really potent interferon response that these cells have to long RNA and actually get gene silencing to be programmed by your short double-strand RNA in cells and knock a specific gene down. And so all of a sudden, functional genomics just exploded with the microRNA pathway using siRNA as a tool. The first example of linking microRNA expression and disease was in 2002 from Carlo Croce's lab, where he observed in press that microRNAs were very frequently associated with regions of the genome that seemed to be involved in cancer, so regions that got deleted or amplified or so on. And that was an interesting observation. And with all of these things said and done, 2003 was a year in which small RNAs were nominated as Breakthrough of the Year by Science Magazine. Business magazines like Fortune saw the money that could be involved in biomarkers and clinical applications of microRNA. And Fire and Mello won the Nobel Prize in 2006 because of their discovery in C. elegans in 1998. The first examples of using microRNA as diagnostics came around 2004 from a lab in Japan, and it was both diagnostics and prognostic biomarkers. And then for the purposes of these discussions, one of the first papers that showed that microRNA was present in serum as a stable RNA and could possibly be used as a biomarker was published in 2008. Just a couple more things, just highlights of the microRNA world. Kaelin's group showed that 50% of microRNA is mapped to cancer regions. Frank Slack's lab showed a direct microRNA target interaction that could cause cancer. microRNAs that could function directly as oncogenes in vivo were shown by Greg Hammond's lab. How microRNAs can influence epigenetics, microRNAs and SNPs either in the microRNA gene or the target gene, and how that could influence their function. And then Frank Slack's lab, again in 2010, showed that they could actually transform cells and cause oncogenesis by expressing a single microRNA. Finally, therapy. So there's a number of potential ways that we can use microRNAs as therapies. We can transfect or provide microRNAs to cells and cure a missing or non-functional microRNA. We can use antagometers, which actually interfere with the way microRNAs interact with their targets, so we can suppress a bad microRNA. And recently there have been papers using microRNA decoys, which are sequences in the that we put in cells that are designed to soak up extra microRNAs with their binding sites. So this is now 2 years out of date. This was a figure from a paper from Gene in which the author described what microRNAs had been nominated as potential biomarkers in different diseases. And what you see is just quite a number of them here, and this was 2 years ago. There's a lot more now. And these, this particular paper is about cancer, and so it's various sorts of cancers that these microRNAs can be implicated as either causal or as a result of the cancer, and maybe be used as a biomarker. So although there's no shortage of papers, and this is a Medline search of microRNA and biomarker, and what you can see is from 2007 on, we're in an incredible growth phase in the number of papers that are published on microRNA biomarkers, and there's now over 2,000 of them, and they're all just a few years old. But there's only a very small handful of microRNAs that are being further developed as assay-specific reagents and maybe ultimately as diagnostic tools. So there's a lot of interest, and it's a very, very new field. So let's talk about this stable extracellular microRNA in blood. Well, we know now, after several years, that these are in the extracellular compartment, that they are incredibly stable, even though there's a lot of ribonuclease in serum and plasma. So they must be protected in some way. And what's known about them now is that a lot of this microRNA is in exosomes or microvesicles, and we'll talk more about those. Some of it, though, is complex with AGO2, so the protein is protecting the microRNA. Nobody's really quite sure how these get into the extracellular environment. It may just be that cells that lyse through apoptosis or whatever are spilling contents in, but they are there. There's also evidence that HDL mediates microRNA transport between cells, and there's also some other proteins that people have found that appear to protect microRNAs. So most things in circulation are covered in proteins, and microRNAs in these cases are not an exception. And you could ask the question of, well, if you're looking to develop a biomarker, does it matter what we look at here? And it depends. All of these are detectable in serum and plasma, but some of them might be more specific for a particular type of disease, for example. So let's talk more about exosomes. So exosomes have been known for many years. People have known that they are budded, essentially, off of cells. In particular, cancer cells can shed large numbers of exosomes, and they contain on their surface and interior proteins that come from the parent cell. They also contain mRNA and microRNAs that come from the parent cell, and large numbers of these can be found in circulation. So one term that's a very interesting one to use about these is the term proxysome. So microvesicles, which contact another cell and they dump their contents into the recipient cell, are acting as proxies for cell-to-cell interaction between the parent cell and the recipient cell, even though these may be in very different parts of your body. So you may have a cancer that's in your breast. It's shedding exosomes into the circulation. Those exosomes can go to a distant site in the body and cause a local immune response, leaky blood vessels, and prime the site for to be a receptor for a metastatic lesion. We also know that exosomes from cancer cells go to the bone marrow and influence bone marrow cells as well in terms of modulating immune response. So this is an incredibly exciting, fast-moving field that holds a lot of promise both for diagnostics and for drug development. So what about the non-exosomal microRNAs? These may still be valuable biomarkers. For example, circulating miR-122, which I'll show you in a few slides from now, can be a marker for liver injury. Circulating miR-1 is a marker for heart injury. Neither of these are predominantly exosomal. These are clearly released from damaged cells into the circulation. So to be thorough, any study that you undertake should look at both whole plasma and also use the exosome fraction. And you may not end up using just the exosome fraction for a biomarker because whole plasma is cheaper, faster, and simpler. And we are developing an isolation kit in collaboration with Exosome Diagnostics that lets you collect both the exosome fraction and the non-exosome fraction of serum or plasma, which will let you decide, essentially, where you get the best signal-to-noise ratio for the biomarker that you're working on. So let's turn to our case study. This was done in collaboration with James Deere in Edinburgh. And this study is on acetaminophen poisoning in humans. And so just for a little bit of background, acetaminophen or Tylenol overdose is one of the most common causes of poisoning worldwide. All that Tylenol that you take is not good for you. And in the US and UK, it is the most common cause of acute liver failure. And the problem is that Tylenol is in everything. It's in every over-the-counter medication you could possibly take. People are not aware of this. They take multiple meds, and they end up poisoning themselves. And what happens is 24 to 48 hours after an overdose, you start showing symptoms of acute liver poisoning. And 3 to 5 days, if it's serious enough, you get complete liver failure, kidney failure, and then you get brain swelling and sepsis and multiple organ failure, and you die. And so many people present with symptoms of Tylenol poisoning. The diagnosis today is one in which they look at Tylenol levels in the blood, and they decide if you're bad off or really bad off, and they treat you accordingly. But there's still this gray area where it would be really great to know which patients are in risk of having really complete liver failure and dying versus which ones are going to recover. And right now there really is no biomarkers for how bad that liver failure or liver damage is. So we asked the question, can we find microRNA markers in circulation of Tylenol poisoning? And then can we use those to distinguish this poisoning from other liver syndromes or a less severe non-lethal case of poisoning? So the way we went about this experiment, we broke it up for you into 3 phases here. Phase 1 is to determine the expressed microRNAs in our population. So there are now over 2,000 microRNAs. Most of them are probably not detectable in your sample. You want to know initially what's there and capture everything that's there and then work from there. So phase 1, we're looking at pooled sample profiling, or you could do NGS of a pooled sample and screen the whole micronome to know what's there. Then we take the microRNAs that are expressed and we profile each individual sample with the microRNAs that are expressed. And so in our experiment, it was several dozen samples, and we screen these only the expressed microRNAs, and they all fit essentially on a 3 to 4 hour plate. And then once you have this data on differential expression, you can build a model. You can then get some statistical power by looking at more samples, including naive samples. And hopefully you end up with a classification model that is between 1 and maybe 12 different sequences, so it can be done fairly readily on patient samples. So here's phase 1. We had 54 plasma samples. 27 of them were controls, so they were normal. 27 of them were patients that had liver injury. We isolated total RNA with a kit that I'll describe a little bit later. And we initially performed QC at this step to ensure that the samples were devoid of inhibitors. So we, in fact, found 1 or 2 samples that were in heparin. So they killed the reverse transcriptase. So it pays, if you're going to do a large experiment, to do some QC in your samples. We then did microRNA expression profiling using the microscript PCR system, which I'll tell you about, and the human micronome, which at the time was 1,850 or so assays. So which microRNAs did we select for further analysis? Essentially, we took anything that was reliably expressed in at least one of the pools, because we weren't quite certain, for example, if there were things that were in only one of the disease pools, whether those are important or not if they're not in the other disease pool. So we took everything, and everything was 377 assays. So in phase 2, which is our secondary screening, we took all of our initial samples, which were 54 plasma samples, so 27 control, 27 liver injury, and we did the microRNA profiling using microscript and those 377 assays along with controls. We did replicate PCR reactions, and we did this on the fluid-ion biomarker. So it took us 4 days. We screened 54 samples with 377 assays. This is essentially 40,000 PCR reactions. fluoride biomarker is great for these kinds of experiments. And we can ask the question, which microRNAs were differentially expressed? So that's just two ways to look at it. One is a scatter plot, and things that are at the bottom are expressed at a low level, things at the top are expressed at a high level. And you can see differentially expressed things are colored green for being expressed at a lower level in the patients, and red are at a higher level. And two microRNAs that I point out here, miR-122.5p and miR-8D55p, are widely known to be involved in liver toxicity and in liver cancer. In fact, the most differentially expressed microRNA was miR-122.5p, which everybody knows about already. But you can see that there are other microRNAs here that are very highly expressed in these samples, which there's no mention of them in the literature so far. So using this data, we attempted to develop a training set. And so we screening a larger cohort is something that we're doing now. But this is an original classifier model that we built and looked at our original 54 samples on. And this is built with the top 16 differentially expressed microRNAs. And so you can see here the positive patients, and there's maybe one that seems kind of suspiciously maybe not really that positive, but these cluster extremely nicely, and the normal controls also cluster extremely nicely. And there are a couple of overlaps here, and we're not quite sure whether these are misdiagnosed or not, or what they are. But in general, even miR-122 on its own classifies these samples extremely well. So what do we do at this point? Well, yeah, we're looking at many more samples. And it's not necessarily true that the strongest changes that you see may be the indicators that you want. So, for example, a microRNA that's very upregulated in a particular disease might be upregulated in a lot of diseases. So it may not be a very specific marker. It's also true that relatively weak changes might add really important specificity. So we haven't yet looked at patient outcome, and once we look at more samples, we'll do that and stratify patient outcome with expression analysis and see if we could find markers that will distinguish between our populations. Obviously, high levels of false positives and false negatives would be extremely undesirable, so we want to pick microRNAs that are specific to liver poisoning and not that are more general markers. And the other things that we would do is we're revisiting the literature with this list of differentially expressed microRNAs. We're using Ingenuity IPA to try to figure out, in a biological context, which of these make the most sense to follow up on, and screening other types of liver injury and other types of samples to help define the specificity. So I want to talk now about the tools that we used in this experiment, from sample collection to data analysis. And the first thing we should talk about is microRNA isolation. So this is for total RNA from extracellular circulating RNA, is what we're doing right here. We're taking CSF or plasma or urine, or we can actually do whole blood, and we're using the miRNA serum plasma kit. And the way this works is we do a chiazol extraction and a bind, wash, and elute. The serum plasma kit includes an assay for a C. elegans microRNA, which you can use as a spike-in to monitor a recovery of your variability in your samples. We typically use anywhere from 1 to a few hundred microliters of input volume. The recovery is fairly linear. We elute in a minimal volume, so we elute in 14 microliters. This lets us concentrate our samples, and the RNA is suitable for all downstream applications. And just as a guide, we're using about 20 microliters of serum for each 384-well array. So you can do a miRNome with 100 microliters. With preamp, which I'll show you in a few slides, you can actually cut it down to less than a microliter. And this protocol, of course, is automatable. The microscope 2 system, which has been evolving and improving in Qiagen for many years now, is a complete microRNA profiling solution from isolation to data analysis. And so I mentioned miRNA serum plasma. If you're using the larger cell samples, we would use the standard microRNA kit. It has a greater capacity for debris. There's a kit specifically to isolate microRNAs from FFPE samples, and if you have stabilized blood and PAX-gene, you can get microRNAs in a PAX-gene as well. The first step would be cDNA synthesis. For either extremely low input samples or normal samples, we're going to do a standard microscript high-spec cDNA synthesis. I'll describe that a little more on an upcoming slide. If the sample is very low input, we would do a preamplification with our integrated microscript preamp system. For either of these in a traditional PCR machine, we use our standard CyberGreen master mix and our free online data analysis tool. I mentioned before that we're using the Fluidigm platform. We have a specific RT that's optimized for the Fluidigm platform, preamp for the Fluidigm platform, and Evergreen master mix, which is great on the Fluidigm platform, and data analysis tool will work as well. So whether you're looking at 96 or a million data points at a time, we've got you covered. So microscript 2 consists of cDNA synthesis, which is a very smart universal RT reaction. So all of the small RNAs are converted into cDNA in your sample. So if there's a new microRNA that's discovered, you can always go back to your sample and look at another microRNA in your cDNA prep. It's a very straightforward workflow from single samples or 96-well format. It's linear from a nanogram to 2 micrograms of input RNA. It's completely unaffected by genomic DNA contamination, and it's extremely specific because of the buffer technology in the kit. We do not reverse transcribe long RNAs into cDNA. The preamplification kit was built to be perfectly integrated into the system. It also is usable on single samples or in a 96-well format. It extends the linear range of microscript down to essentially as low as you can go. A single cell level input to look at a miRNome. It's very reproducible, very linear, and for body fluids in particular like urine, it's essential. So rather than purify RNA from a liter of urine, you can use microliter volumes. In our real-time quantification system, we have a number of built-in controls. The spiking, which I mentioned, we have normalizers that are added onto all of our arrays. And then there's RT controls for RT efficiency and PCR controls for PCR efficiency, and these help monitor for inhibitors that might be in your samples. All of our assays are extensively wet lab validated. We're current at the moment up to miR-based 19.5. We provide assays, focused arrays, the whole miRNome, custom arrays, essentially anything that you need we can make for you. And all of them are usable with our online data analysis tool. This is just a slide showing actually the miR-based version 16 miRNome. We have miRNomes for human, mouse, rat, dog, and rhesus. All the assays are validated. Every array is quality controlled. And we also can do other species if you're interested. The focused arrays are something that we shine at. These are different cancer type specific, different biological function specific. This has gone through literature mining, data mining, all available public information to help build lists that are biologically relevant to scientists that are studying these different things. This is what an array looks like, just so that you can see. This is a high content array. It's 372 different assays along with controls. This is the way the MyFinder 384 looks. It's the 372 most abundantly expressed and best published microRNAs in miR-base. It's also how the serum and plasma array looks, which is all of the microRNAs that we can detect in many, many, many serum and plasma samples out of miR-base version 18 without preamplification. So this is just a couple of examples I want to show you of microRNA profiling in serum. This is the high content 3 to 4 well array. So we start with 200 microliters of serum. We elute our sample in 14 microliters. We take a tenth of that, so it's 20 microliter serum equivalent, do a high spec RT, and run it on the array. And this is without preamplification. We see lots and lots of nice strong signals. When we compare cancer versus breast cancer versus normal, we see some microRNAs that are upregulated in breast cancer, and a few of these are published in literature as being important. So urine. Let's talk a little bit about preamplification. So we are using 200 microliters of CSF for urine for our profiling. We isolate our RNA the standard way. We do an RT essentially the standard way. And then we take an aliquot of that RT reaction and use a preamp reaction, which is specific for the assays that you're interested in. So every array that we sell has a panel of validated assays in a preamplification reaction. So it's a multiplex reaction. It can be 5, it can be 96, it can be 384. After preamp, we delete the sample, do real-time PCR. And so it's this one step that's built into the middle of this preamplification, where the middle of this workflow where we're adding a preamplification step. So again, RNA to cDNA, cDNA preamplified, mixed with master mix, spread on an array, run the samples, do data analysis. The panel on the right is just an example of 2 different operators going through this process on replicates of the same sample, and you can see that they get essentially the same data. Urine, we do a mandatory preamplification. Again, we're only using 20 microliters of urine for this profile. This is just an example of 2 independent experiments on the same volunteer, and you can see that the results are very reproducible. And we see a lot of microRNAs. When you look at 2 different people, and I'm not going to name names to tell you who these people are, you can see that, in fact, their profiles are different from each other. They generally follow this trend, but there are a few outliers, and it certainly doesn't look the same as when you use replicate samples. So urine is not a problem. Small volumes of urine are not a problem. Rat, mouse, not a problem. So this is CSF. We're, again, using 1/10 of the prep, so 20 microliters of CSF. What you see on the left here is a comparison of replicate isolations and replicate profiles, which fall very nicely on the line. Things at the high end are expressed at extremely low levels, so they're a little bit more noisy. But if we look at 2 different people, and by the way, we bought these. We didn't actually do spinal taps on people in the lab. You can see that they're actually quite different from each other. They all still sort of fall on this line, but there are ones that are expressed more highly in one person versus the other. And we're doing some profiling in CSF now. They're hard samples to come by, so we don't have, at the moment, any data on, for example, any people with diseases that you might want to monitor by looking at microRNAs in CSF. I want to round up the technical part of the talk talking about data normalization. So we do data normalization in a number of steps. The first one is on every array that we have these controls. So we're looking at what's called the miRTC and the PPC. And this tells us, or tells the software that's doing your analysis, whether or not there's significant inhibition in your sample, whether you can trust the data or not. So this is extremely useful for particularly biological samples like urine and serum and plasma. And so that's sort of automated in the system. It's great to have those. Every array also has SNO RNA controls. These small RNAs are expressed at very high levels in cells, but in extracellular fluid, this is serum, for example, you can see that the expression level is extremely low and they're extremely variable. So they are not in exosomes, and there's just little bits of this debris floating around. So we don't normalize against these, but we use them as an indicator of whether we've got severe cellular contamination or not. So if you compare the CT values of these in blood to serum, you can see that there are some of them that are expressed extremely highly in blood, like RNU6, and extremely low in serum. And the delta CT between these is 12.5, which is 10,000-fold difference in abundance. So if your CT values for these are well above 30, you're generally fine. And so it's just something that's there, and it's something reasonable to look at. The real data normalization is here. So when we do data normalization, the first thing we do is we normalize to the RNA recovery control, the spike and C. elegans mimic. And then if the array is unbiased, in other words, it's an array of all the microRNAs that are detectable in serum. It's an array of the top 384 microRNAs. It's an array of the whole micronome. We normalize to the CT mean of the commonly expressed targets between our samples. So we look at all of the microRNAs that are reliably quantified in the samples, and we get the mean of those, and that's what we normalize against. That works just fine if you're looking at a reasonably sized population. If you're further down your experiment and your array is biased, so I'm only looking at a small panel of microRNAs that are of interest to me because they're regulated in my system, what we then would do is from the earlier experiments where we're doing the initial screening, pick several invariant microRNAs and add them to this new array and use them as normalizers. And we find these using a program like NormFinder. All the data analysis happens on our website. You can actually also download Excel templates and do it on your computer if you don't want to upload data to our website. Does all the analysis, all the plotting, all the graphs are all done, and this is accessible to anybody. So I just want to leave you with just maybe a little bit of teaser about the other webinars that we're giving. We spoke about a little bit about microRNAs, really not very much, and the kits that we have available for isolating microRNA. We didn't really talk about automation. We didn't talk about the service core here. We did talk about microscript, but we didn't talk about precursor assays, which are very useful for microRNA dynamics in the cell. And we didn't talk at all about functional studies. So you'll hear more from Sam about how to transfect microRNA and reporter constructs into cells, how to use mimics, inhibitors, target protectors, and so on. All of our gene-specific resources at Qiagen are found at qiagen.com/geneglobe. So you search for a keyword, kinase, cancer, breast cancer. You search for accession numbers, you search for gene names, and it will show you all of the gene-specific components that we sell, whether it's an mRNA assay, a microRNA assay, a Biomarker assay, whatever it is, they'll all show up there. The other thing it will do for you is show you which pathways those microRNAs or other genes are involved in. These are all clickable, so you can actually look at each of these gene products and look at the products that are available for different gene products. And with that, I'd like to say thank you. I'm going to turn the talk over so that we can field some questions. Again, thank you for coming.